Okay, let's get started then. So this lab is on reproduction and development, and you'll be doing several exercises in the lab. You'll combine what you already saw in the first lab in this section, which was when you looked at the rat. You looked at both male and female gross anatomy, the very first lab that we did in this series of exercises. So I will talk about some of those organs again, such as seminal vesicles, or the ovary, or fallopian tube, etc. Okay? So you'll need to make sure you know the material from the first part, that first lab. As you know, lab exam 2 is next Wednesday night, May 1st, 8:20. There'll be handout on B-Space. Make sure you come on time. So be seated, ready to go by 8:10. We want to hand out the exams in Scantrons. The exam will indeed be multiple choice format. There will be up to 10 choices per question. So the way you've seen this before, so the way this is going to work. Okay. Trust me, this is so much easier than the past format where you had to write in all the answers, okay? And it was always the case where you had to take and put in all the correct answers, okay? So the way it's going to work is question 3, let's say, we'll have A through J, and then they'll have a little A, a little B, whatever, and then you'll bubble in. For some of the questions, you'll bubble in only one answer, A through J. In others, you may need to bubble in more than one, and we'll say that specifically, okay? So if there's only one answer, you bubble in one, okay? So any questions about this? This is only about, yes, question. In some cases, yes. Okay. The question's only worth typically 1 point or half a point because what do we do? Oh, you get 5 of the 6, and it was worth half a point, so each one is then 1/5 of a half point. It's just not worth it. So half point. So it's usually full credit only. Okay. So no subset within there because it doesn't really make sense. It's only half point or 1 point for the question. In the back. No, the entire test is multiple choice. Entire test. No written in answers. No drawing cladograms. None of that sort of stuff. Question. Say that again. So the question was, before it was multiple choice, was all or none? Yes. Yes, it was all or none in the past as well. You're not being treated any more harshly than other semesters. And in fact, I've had students from past semesters already email me complain that they took the format in the format they had. Okay. So anyway, let me just show you one of these. Okay. So this would be it. And the way I'm doing this to not bias the answers is probably the fastest way to do a given question is to think what is the correct answer. Okay. And then I have a table like this, and I will put them in alphabetical order. Okay. So if you're looking for some answer that ends with H, you'll find it there. Okay. So I will always try to put the answers in a table in alphabetical order because that will make it much faster for you than randomly looking for where's Anthozoa or Parazoa or whatever. Okay. Does that make sense? So that's again just an attempt to make it go faster. I think you should have plenty of time. Okay. This is far easier format than where you used to do a station and rotate through for a minute 45 seconds and freak out. Okay. So any questions about this? It's only about grades and format right now. It's not about the learning, right? This will still allow you to show us that you learn material. All right. Asexual versus sexual reproduction. As you know, asexual genes come from one parent. So gametes are not produced through meiosis. You don't get that genetic shuffling, so you don't get the generation of genetic diversity. Whereas with sexual, you fuse gametes. Not necessarily always one end, but with the animals, most of the animals are indeed diploid in the adult form. There are some mammals that are tetraploid. So anyway, sexual versus asexual. Asexual disadvantages. It lacks that genetic diversity, but there are tremendous advantages. You don't need to find a mate. So if you live in isolation, you can still produce more members of your species, and they're genetically identical. And so you have a quicker generation time, and successful genotypes are maintained. So if you're already in that stable environment, as your offspring are genetically identical, you maintain a genotype that's suited for that environment. Of course, if the environment changes, that may not be very good. So if organisms have the ability to do sexual versus asexual, we usually find asexual forms of reproduction in a stable environment and sexual forms in an unstable environment. So the disadvantages of sexual reproduction: mate selection and timing. You have to be ready to mate at the same time as the other sex is ready to mate, right? And they have to be receptive to your advances. The number of gametes required is quite large. In a given ejaculate in humans, maybe 300 million sperm. Typically, 2 or 3 might be. A would actually fertilize a secondary oocyte to form twins or triplets. So not real good rates there. And you only have 50% of your genes in that offspring. But of course, the advantage is that genetic diversity, and you know about monoecious versus dioecious. And in those organisms that are monoecious, they can sometimes require cross-fertilization. So that means, unlike pea plants, which can self-fertilize, you can have eggs and sperm produced by the same individual, and they can undergo fertilization events into offspring. That's self, whereas there are mechanisms to promote cross-fertilization, so it's egg and sperm from different parents. And that, of course, promotes that increase in diversity. And this graph just shows you how a given population that has asexual reproduction can increase their numbers faster than those that have sexual reproduction. Any questions? And in terms of parthenogenesis, now you typically won't have sperm give rise to another offspring, and what would be the basis of that lack of that ability? What's that? They could still have organelles, they could still have mitochondria and things like that, but did you say fewer? Fewer. Yeah, so you have less cytoplasm, less nutrients, things like that, okay? But they still have organelles, all right? We've talked a little bit about the bees, where the males are one end and the females are two ends. So the males are produced because those eggs aren't fertilized, and they give rise to the males, and then females are two end where sperm and egg have combined, and the workers are sterile. They are not able to produce eggs themselves, but the queen can. And they both start off diploid, but what happens is, during development, the bees that become queens are fed royal jelly. You may have heard of that before. So it's a dietary difference that results in the females being queen or workers, okay? And some fishes, things like that, they can change their sex, things like that. They can have parthenogenesis. If you have reproductive season and you have that sexual reproduction, you have to time gamete production so that both sexes are willing and ready to mate. And you typically want the offspring to be born in a favorable time of the year that those offspring can then develop, right? So you need to have environmental signals. Usually it's like daylight, length of daylight. You certainly have noticed now that spring is here. Plants have opened the flowers. Again, plants have to do just like animals. They have to time when they release their gametes. There could be things like changes in salinity if it's an aquatic organism. Various things like that, temperature changes. Okay? And as I mentioned, some things can alternate between sexual or asexual. We've seen this. What is the smallest group that contains this organism? Anemone? Anthozoa. So next week on Monday night, I've already prepared the lecture slides for you. They're in your reader. They're 6 per page. I'll go over this. I'll highlight key things, ask you to identify structures, and come next Monday night, the answers will be coming very quickly. So I know you can learn a lot. Please don't wait till Tuesday to try to do this. It's just too much material, okay? But this is a situation where you can just put a lot of effort in and probably do well in lab exam too. It is a lot of memorization. I am trying to get some conceptual questions in there as well, okay? So for example, here's a conceptual question that I thought of right before lecture. So you know that for humans, and we're going to go over this, that when the female ovulates, it's not a true ovum that's released. It's a secondary oocyte. And we'll see this secondary oocyte is run by a lot of cells. We'll go over that. But if you have that secondary oocyte, there's a bunch of cells around it, the granulosa cells that are released with it. So it's not just the secondary oocyte that gets released. There's this layer of zona pellucida around here, which is an extracellular matrix. There's, of course, the egg membrane, there's the zona pellucida, and then there's these follicular cells around here. And those happen to be granulosa cells. This entire structure that consists of the secondary oocyte of these cells is typically just called the secondary oocyte. It's a little confusing. Biologists should be a little bit more careful when they use this term. So when we talk about a developing follicle, we are referring to oogenesis and what's happening to the actual oogonium and the cells that surround it. So we can talk about these as being follicular cells, okay? And as you know, you go through that first division, and you yield a secondary oocyte and a polar body. And these are 1N, 2C. And then the second division, you know, who knows what happens here, yields the ovum nucleus and a second polar body. So strangely enough, there are sometimes, it's very, very, very rare, that we get what's called semi-identical twins. And that is, we have a sperm, and they fuse what we think is this secondary oocyte here to trigger that event. And then this polar body that's produced here is also fertilized. So as a result, the genetic material in this should be fairly identical because it was a separation of sister chromatids. So a theory type question would have been, I could have asked you this and said, you know, would it have been this polar body that had the potential to do this, or this one to yield offspring that had very similar maternal DNA? And it wouldn't be this one, because in the first one you separate members of homologous chromosomes, right? So you would expect vast genetic differences between this secondary oocyte and this first polar body because you separate members of homologous chromosomes. Whereas here you're separating the sister chromatids, right? So that would be a theory type question. I would have to develop it for you a little bit. So I can indeed get theory type questions in this, even though you might think it's a lot of memorization. And there will be. I certainly have the identified structures, the analogous structures in other organisms, but I'm going to definitely try to get some conceptual questions in there as well. All right, so here's an example of asexual fission like that. Here we see budding asexual reproduction there. What's the smallest group? Hydrozoa. And by the way, when I'm asking you questions on the lab exam, like what's the smallest group, please don't think about, oh, there are bacteria on that surface of that I can't see, and he's making it into a trick question. It's not that at all, okay? Ignore parasites, go for the big thing visible, okay? All right? Clearly there are all those other microscopic things that we're going to ignore, okay? But I have had students say that they did that. They don't, it's not trick questions, okay? Here shows you regeneration in that starfish. You can see this is that original arm had its small part of the central disc, and it's regenerating here. In terms of this, I'm not going over this much. Dr. Patel spent a fair amount of time talking about this, how we have these whiptail lizards where they're all female in the population, and they act female-like when they have high levels of estrogen. When they have low levels of estrogen, they act male-like. The male will mount the female, pretend to copulate with it, and that actually stimulates egg release from the female here. So you still need some mating behavior, even though they're only all females. And what happens there is when she makes the eggs, they're haploid, but then they just undergo another round of DNA duplication to make them effectively to see the typical two sets of information, okay? Is that clear? And Dr. Patel went over that quite a bit. You want to go over that again? Yes, no? Yes, okay? So we can just go back to this. So if we start off with a cell here, I'm going to use 2N in parentheses, so we have that female. She goes through the first normal division, 1N, 2C, 1N, 2C. But remember we had S phase in there, so we've gone over mitosis and meiosis a lot. All right, and then we do the second division like this, which effectively makes 1N, 1C, 1N, 1C, 1N, 1C, 1N, 1C. Typical sort of thing. But if we need a equivalent of a diploid, two sets of chromosomes, we can go through an S phase, and we don't get nuclear division, so they all remain in a common nucleus when we reform the nuclear membrane. So this effectively becomes like 2N, 2C. I put this in quotation marks because this, of course, is identical, right? Each set's identical because you only have one set to start with. So when you duplicate it, except for errors in replication, it's effectively the same. So there are two sets, but they happen to be similar because of the way it worked when the S phase occurred. All right? And Dr. Patel went over that quite a bit if you look at that notes. And for the 1A exam, it is indeed comprehensive, okay? So that was intent to try to reinforce some things. In terms of the cycle here, we're going to talk about gametogenesis, which is the production of gametes both in males and females. Then we're going to talk about fertilization. And if you could write this down now, it will be very helpful. When we talk about fertilization, we're going to talk about two separate events of fertilization. We're going to talk about membrane fusion. That's where the sperm membrane fuses with the egg membrane. There are receptors involved. And the second aspect of this is what's happened to the DNA. Okay? So there may be DNA fusion to form that true 2N cell. Okay? And hopefully that will help you clarify when we talk about humans, because it's a little complicated humans. Question? Yeah, do you have a question? Yep. Okay, well, so this polar body is genetically very different from this polar body, and these two are very different, right? So if you think about it, so here's a chromosome that came from mom, here's a chromosome that came from dad in that female. And they're not the same because her parents weren't the same, right? So when she duplicated like this and does this sort of thing, when we first get to this part, and we get the 1N, 2C like this, for those 23 types of chromosomes, they're very different, right? So this cell is genetically extremely different than this one, because for each of those 23 we get different ones. Does that make sense? Yes, no? Okay? And then, so when we go down here and we had this 1N, 2C, these two are genetically identical for the most part, because all we're doing is separating sister chromatids, right? The differences are, though, there are some crossover events, okay? The crossover events may mean that some of these chromatids are slightly different. So these two are extremely similar, and then these two, if this underwent, this would be very similar. So it would have to be this second polar body here that underwent the fertilization to have the two be the same. It wouldn't be this one and this, because those two are so genetically different. It potentially could have been this and this. I don't actually know if these divide. Okay, so it may potentially been that, okay? But I think almost everything I've read says when it does happen, which is very rare, it's this ovum nucleus and probably the second polar body, okay? Make sense? Okay, question? So does the offspring actually develop? Not very far in development. These things are usually aborted, but when they go back and do tests later on, they see that they're genetically similar. Yeah. So probably 50% of all conceptions post-implantation are aborted due to genetic defects. The person may never even know she was pregnant because it happens so early in the developmental process. Okay. So yeah, so this is a situation where probably most of those get aborted early on, and all those other genetic defects, but in a few cases it goes far enough along that they have delivery or C-section, and then they do genetic tests on the individual. Okay. That answer it? Okay. All right. So we have the fertilization. Cleavage is a unique series of divisions. And I'm just illustrate it here. So what happens is we start off with an egg cell like this with a membrane around here. Okay? We have a fertilization event, and then we start doing very rapid mitotic divisions. We have S phases in there. but we don't have any cell increase in size. So that's very unusual because effectively if you do this long enough, the cells disappear, right? They get smaller and smaller, so it's called mitotic oblivion. But what happens is these cells are smaller now, and then they can migrate, and then you can form a multicellular structure. So cleavage refers to a unique set of mitotic divisions where you're not getting increase in cytoplasm before you undergo that cytokinesis, before you undergo mitosis. So we'll see when we look at this that we really don't change the volume of that egg cell. Just get smaller and smaller within there, okay? And you'll see that. Gastrulation refers to cell migrations to form 3 germ layers, for those that have 3 germ layers, okay, triple-plastic organisms. And we'll see that they form the endoderm, the ectoderm, and the mesoderm, okay? And then we'll talk about neurulation, certainly in the chordates, so we'll mostly focus on. We'll have neurulation forming the dorsal hollow nerve cord. Larval and juvenile forms, these are forms that cannot produce viable gametes, okay? For humans, we call them adolescents. There's no real metamorphosis in humans per se, but then we get to adults to make viable gametes, and then we repeat the process, okay? So we'll be looking at several things in lab. So I'll write the organisms that we're looking at in lab. I've tried, in the past, I've written this where you first look at one organism, then another, but then people got really confused about the chronology of events. So now I tried to organize it by chronology. So that outline that I did before is the chronology that we'll use, but some organisms allow us to look at things easier than others, okay? So what we're going to talk about is gametogenesis. So we'll have you look at cross-sections through an ovary and the testes of a rat, so you can look at gametogenesis in the 2 sexes. We also have some slides from frogs or ovary that you can look at as well. We'll talk about gametes that are produced. Now, we'll actually have sea urchin gametes. So we have sea urchins. We've injected with potassium chloride, which causes them to release their gametes. We can't sex them ahead of time. We don't know their gender externally. So what we do is we end up, we may have to inject several of them, end up with some males and females. So in lab, you'll have sea urchin sperm and sea urchin eggs. They'll bring them out to you in lab, and you're going to use plastic pipettes. And then you have a depression slide like this. I'm going to go over this now and hopefully remind you again. Depressions like this. And you're going to put the drop of some eggs in there. And you'll look at them, and you'll see that they're evenly spaced, but they don't appear to be touching. They have a jelly coat on the outside, and we'll talk about that, which is the same index of refraction as the seawater. So it's really hard to tell. It's there. Sometimes we get some species where there's pigments in the jelly coat so you can see it, but there's a jelly coat out there that helps protect these. So what you're going to do is put these in here. You're going to look at them with the microscope. And then you're going to go over and grab a pipette with some sperm. Then you're going to go back to your bench and add some of that sperm to this. Then just put that pipette with the sperm in the waste beaker on the bench. Do not go back and put it back with the other containers. And what will happen is the sperm will be released and start to fertilize those eggs. And that will happen in less than a minute. So you'll want to watch it the entire time that you do that, and then we'll look for certain events, okay? So we're going to look at the gametes from the sea urchins because we can actually get fertilization that occur. We'll see some aspects of that. And then we'll see some cleavage reactions in the sea urchins, maybe to the 2 or 4 cell stage, depends how warm it is. Maybe 8 cell stage at most, but that's about it in the 3-hour period. Then we'll look at gastrulation. So we have, in terms of the urchins, they are bilateral symmetry in terms of the larval forms. And we won't talk about neurulation with the urchins, of course, but we will look at frogs. So we'll look at some preserved examples. We'll have a frog ovum. We'll see different stages, 2-cell, 4-cell stages. We'll see stages within here called a morula, a blastula, a gastrula. And then we'll see a neurula stage. So it allows us to see those stages, okay? And then we'll also have you look at chicken egg, and you can see developing chick embryo, okay? So that first image that I showed you here was of a human. It's about 6 weeks old. When you look at chick embryo, which are about 3 days old, you'll see they look very similar. So clearly, human development is slower than the chick, but they look very similar at that stage, okay? So in terms of sexual reproduction, gonads are the primary sex organs. They make the motile gametes in case of the testes, the sperm. The ovaries, few, typically non-motile, all right? And of course, they produce hormones that you've heard a lot about, okay? Accessory sex organs, they play a role in gamete viability, gamete delivery, maintaining the developing embryo, et cetera, okay? So those are this, and we've given them names sometimes like seminal vesicles, Croydon glands. So I'm going to go over some of those, okay? Here shows you for spermatogenesis. Here shows you a testes here. Since it was Easter recently, this looks like a testes here. Of course, there'd be 2 of them typically, at least in humans. You open it up, and what you'd find in here, as Dr. Firestone mentioned, would be like a bunch of spaghetti. And in fact, each of those pieces of spaghetti would represent a semester tubule. There'd be about 750 of them in there. Okay, so there's a lot of semester tubules in that one testis, and you're making lots of sperm. So in fact, if we set a timer, I'm going to set it for 10 seconds. This is an experiment for the males in here. So the females are in this 10 seconds. Not much is happening in them with respect to gametogenesis. And in the males, that's done. We've made about 100,000 sperm in that 10 seconds. So the question becomes, this is always the question people want to ask, but they never ask, what happens to all those sperm? No, we don't explode, okay? We just reabsorb them, not a problem. The body is amazing at recycling things. So if they're not released, you just reabsorb them, okay? It takes about 20 days to make those sperm. And as you know from the 1A lecture Dr. Firestone had about 3 hours to go over this. So I'm going to do it in about 10 to 15 minutes. So I know it's going to be fast. We have, in one of these tubules here, remember that there are cells outside called the interstitial cells. They are Leydig cells. I'll show you an image of that shortly on actual cross-section. Here's one of those tubules. Remember there's these cells that are called Sertoli cells, and they basically form a blood barrier within the testes. So this is one tubule like this. You can think of those Sertoli cells like this. I'll draw one in more particular detail like this. So we have the spermatogonia here, and as they go through this process of DNA duplication and meiotic divisions, they effectively get sorted to this side. And what happens is, so the primary spermatocytes will be on this side, eventually, across that into this lumen. So basically we have something like this. If there's the tubule, here's the lumen, there's a barrier provided by those Sertoli cells. So what gets across there are the primary spermatocytes.